A very good morning to one and all who have gathered here. I'm happy to see all the who have come here to present their ideas and their research techniques which they have carried out. And it has been a long time since we conducted the Food Fest and the Biothrough conference, and now we are going to build up the gap with the Biotekionz 2020. I need to thank, first of all, Dr. Saira Meshar, who initiated this event a couple of years back, and Deputy Dean Yuvraj Sir, who is in public-private partnership now, and our HOD Gnanasekharan Sir, and other staff who have supported us to make this event to conduct now, and that's why we are in. Let me read out the rules. You will be given a presentation timing of 10 minutes and 5 minutes of questionnaire session. So please make sure that you finish within your time limit. And without any further ado, let me call the first team. Vrinda and Yamlata being guided by Ramya ma'am. Every one of us would have got time or other. But yes, sir.
Hira.
I'm going
here. The evaluators are there, I guess. 2 evaluators will be there.
Udayamati ma'am.
Oh, let me introduce.
Yes.
And let me. Sorry, sir. I didn't. I have to introduce Anusha ma'am and Udayamati ma'am. Hello, sir.
Yes. They are there inside. Just welcome them.
Yeah, sir. Yeah. I'll just. I'm happy to see our professors who have come to judge the students in their ideas. I welcome Udayamati ma'am and Anusha ma'am in the judge panel. Let us start with the competition, the oral presentation. Let me welcome the first team, Vrinda and Yamlata, being guided by Ramya ma'am. Every one of us would have got hurt sometime or other, but with time they get healed off. Now let us have their presentation on the title, Wound Healing Process. Over to Vrinda and the team. Vrinda, you can keep moving it. So let us welcome the second year student, single girl team, Kinsa Fatima, being guided by Yuvraj Sir, who is not scared of COVID-19. We are expecting the vaccine soon. The team says that they have an alternative. The title is Mesenchymal Cell Therapy for COVID-19. Over to Kinsa.
Is my screen visible? Hello?
Yeah, Kinsa, I can.
Yeah.
All the best.
Thank you. Hello everyone, thank you for having me. My name is Kinsa Fatima and I am going to tell you about the mesenchymal stem cell therapy for COVID-19. First, let me give you an introduction. Almost 1 year ago, the novel coronavirus cases started. After a few months, it became pandemic. When people started dying because of coronavirus, I started to wonder how people die because of a virus. Then I came to know all the details about coronavirus. I thought there must be a solution to save people from coronavirus. Then when I came to know about the last 8 symptoms of coronavirus, which are nothing but increase in nucleophile count, which are white blood cells, and cytokine storm. So first let us see what are mesenchymal stem cells. Mesenchymal stem cells are an example of tissue or adult stem cells. They are multipotent, that is, they have the capacity to self-renew by dividing. Mesenchymal stem cells are also known as mesenchymal stromal cells or medicinal signaling cells. They can be differentiated into various types of cells like bone cells, cartilage cells, muscle cells, etc. etc. Okay, so the main source of mesenchymal stem cells are bone marrow. It has been frequently used for research purposes. These bone marrow stem cells do not contribute to the formation of blood cells, so do not express the hemopoietic stem cells markers. Okay, so left side picture indicates the mesenchymal stem cell culture process, and the right side picture is a microscopic view of mesenchymal stem cell. Now, moving on, mesenchymal stem cell therapy. Mesenchymal stem cells are multipotent as we have discussed. So, this technique is being clinically explored as a new therapy for treating a variety of immune mediated diseases. Mesenchymal stem cells express a variety of chemokines and cytokines. Cytokines are nothing but a large group of protein, peptides, or glycoprotein that are secreted by specific group of immune system. Chemokines are nothing but a classification of cytokines based on their behavior, structure, and all that. Moving on, mesenchymal stem cell express a variety of chemokines and cytokines that aid in repair of degradative tissues, restoration of normal tissue metabolism, etc. Mesenchymal stem cells sources are not only just bone marrow, umbilical cord, amniotic fluids, many more sources are available. How mesenchymal stem cell therapy is useful for treating COVID-19 patients. There's no standard treatment for this disease up to date. Probably preventing the severe acute respiratory infection, that is a form of COVID-19, as the most dangerous phase of this disease, can be helpful for the treatment and reduction of the death rate. Mesenchymal stem cell therapy can reduce the number of neutrophils, cytokine storm produced by the COVID-19 infection. In a number of cases, the administration of these cells has been beneficial for COVID-19 patients. Also, mesenchymal stem cells may be able to improve the pulmonary fibrosis and lung functions, which are caused damage by the COVID-19. Okay, moving on to the major symptoms of novel coronavirus. As we know that most common symptoms which we know are fever, dry cold, and all that. There are serious symptoms which is the difficulty in breathing or shortness in breath. Now, increased studies indicate that cytokine storm may be contributed to the mortality of COVID-19. That is, the death of the COVID-19 patient is mostly caused by the cytokine storm. An early indication of severe COVID-19 is increased number of circulating neutrophils in the blood. Mesenchymal stem cell therapy can be used for decreasing the number of neutrophils, reducing the cytokine storm, reducing the difficulty in breathing. So, mesenchymal stem cells can be effective for the treatment of people for affecting COVID-19. Okay, this is a picture which represents what is cytokine storm. When COVID-19 virus attacks our T cell, B cells, macrophages, it releases some cytokines and chemokines like IL-2, IL-7, IL-10, GSCF, IP-10, MCPI. These all are nothing but cytokines and chemokines. IL is nothing but interleukin. CRPF is C-reactive protein. TNF is tumor necrosis factor. These are all cytokines. When mesenchymal stem cells are given to that particular cells like T cell, B cells, macrophages, uh, when the IL-2, IL-7 are released, they cause damage to the lungs. These can be prevented when we give mesenchymal stem cells to that cells. So, what is cytokine storm? A severe immune reaction in which the human body releases too many cytokines into the blood too quickly. Cytokines play an important role in normal immune responses, but having a lot of them released into the body all at once can be harmful. A cytokine storm can occur as a result of infection, autoimmune condition, or other diseases. Mesenchymal stem cells has been shown to possess a comprehensive, powerful immunomodulator function. Cytokine storm treatment with mesenchymal cells. Preventing and reversing the cytokine storm may be the key to save the patients with severe COVID-19. This study aims to investigate whether MSC transplantation improves the outcome of patients with COVID-19. The intravenous transplantation of MSC was safe and effective for patients with COVID-19, especially for the patients in critically severe stage. Mesenchymal stem cells can reduce the cytokine storm infection. In a number of studies, the administration of these cells has been beneficial for COVID-19 patients. Also, MSC may be able to improve the pulmonary fibrosis and lung functions. Okay, so there are many clinical trials going on in China, America, Japan, and all the places. So a few cases I'm going to show here. In a case recently in China on a female patient who was affected by the acute COVID-19 syndrome, when she was treated with umbilical cord mesenchymal stem cell therapy, she recovered in 21 days after the transplantation. Then again a case in which 65 years old female patient was diagnosed in a critical condition with COVID-19. The patient had an increased neutrophil count of 87% and lymphocyte decrease of 9.8%. The patient was treated with umbilical cord mesenchymal stem cells alone. The results of the study showed that after the second injection, all that is increase in neutrophils, lymphocyte decrease, all become gradually proper. The patient was removed from the ventilator and was able to walk. The number of white blood cells and neutrophils in the patient decreased to normal while the number of lymphocytes increased to normal. These results suggested that umbilical cord mesenchymal stems could be ideal treatment option alone in combination with other immune modulators. In another study taken place in China in collaboration with the United States, 7 patients were treated with COVID-19 from January 23 to February 16. All the patients improved significantly 2 days after the stem cell therapy. The patients, okay, so the clinical symptoms of all patients improved significantly. What are the results of mesenchymal stem cell therapy for COVID-19? After the transplantation of mesenchymal stem cell therapy, the person who is treated gets an immunomodulation, that is increase in his immunity. If there is a repair in his tissue that gets repaired, it protects the organ from further damage. It reduces the cytokine storm caused by the COVID-19 virus, inflammation of lungs, shortness in breath. All this can be reduced. So my conclusion is that it might be noteworthy to test the safety and efficacy of MSC transfusion in COVID-19 patients, especially for severe or critical cases. The mesenchymal stem cell therapy does not have any other adverse side effect on patient, thus making it as better treatment. Mesenchymal stem cell therapy offers a promising approach towards mitigating the deleterious effect on infection in COVID-19 patients. Thank you. Okay.
சொல்லுங்க எங்க இங்கயா
Ask your clarification or any question.
Sorry,
Yeah,
ma'am,
okay.
I can't.
Kinza Fatima.
Yes, ma'am. Good morning.
Yeah, it was good morning. It was a good presentation. My first thing is the doubt is okay. I know the stem cell therapy, but in India, the initial stages of stem cell therapy itself is not so far success. Do you agree?
Yes, I know that, but they can try on the patients who are going to die because of the coronavirus, ma'am.
My first doubt is identifying COVID-19 in acute state itself. It's being difficult. So by the time, if we are identifying and if we are transplanting, how much possibilities is there? The patient, by the time the patient may get severe condition also after transplantation. But how come this mesenchymal stem cells, by producing these cytokines, how come?
Yes, ma'am. We can identify the severe acute COVID-19 virus at an early stage by knowing the increase in the neutrophil count, ma'am.
Yes, but comparatively, the symptoms what we are most of the cases we are seeing is asymptomatic, right? But in severe stages like the old patients only, we are able to get uh, the severity only we are knowing by the chances of uh, getting infected by other means, like the symptoms வேற ஏதாச்சும் சிம்டம்ஸ் தெரியுதா அப்படின்னு பார்த்து தான் அந்த சிவியாரிட்டிஸ் நம்ம வந்து ஐடென்டிஃபை பண்றோம் இல்லையா? ஸோ அந்த என்னோட கொஸ்டின் அதுதான். ஸோ அந்த நம்ம ஐடென்டிஃபை பண்றதுக்குள்ள ஸோ தட் பேஷண்ட் வில் பிகம் மோர் சிவியர். ஸோ பை த டைம் இந்த டிரான்ஸ்பிளான்டேஷன் பண்ணும் போது what is the possibility of getting cured? So and again this stem cell therapy is one of the costliest treatment. Yes, compared to others. Normal transplantation, organ transplantation compare பண்ணும்போது this stem cell therapy is huge cost. So spending on that, so அதுக்கு ஒரு particular use இருக்கணும் இல்லையா? So spending so much of money on that, so how can able to there are possibilities of getting failure also, and the severity of cases பொறுத்து.
Yes, ma'am. There can be failure, but when it comes to a life, they can just. In America, all they have done many clinical trials, ma'am. In that, almost 90% of the cases has 90% of the patients who underwent the mesenchymal stem cell therapy were saved from dying, ma'am.
Okay. So how far this study, in how many patients these studies have been carried out? Can you tell me that?
Mostly in America and China only it has been carried out, ma'am.
Okay. Can you tell me any exact number?
Exact number I don't know, ma'am. Around they have done a 20 trials, I think, in America, ma'am.
Okay.
The 20th trial was taken a few weeks back, I think, ma'am. In that 20 patients were admitted in hospital, ma'am. 10 were given 10 were treated with some different mesenchymal stems and 10 were treated with some other different mesenchymal cells. In that, all the 19 got okay and walked off. One is still in a severe case. I studied somewhere, ma'am.
Okay. Can you go with the second slide?
Second slide, ma'am.
Yes.
Time எடுத்து she is personal attack.
This one, ma'am? Or this one, ma'am?
Yeah. No. The previous.
This one.
So you are telling the bone marrow will be the source of this mesenchymal stem cells, but how these bone marrow cannot able to contribute to the formation of blood cells?
Sorry, ma'am, I can't get you, ma'am.
No, in the last point you are telling it is the original source. Bone marrow is the original source for this mesenchymal stem cells.
Yes, ma'am.
And how we are telling that bone marrows will not contribute in the formation of blood cells? We know actually bone marrow is the main source for this hematopoietic stem cells. From the hematopoietic stem cells only, all the blood cells are getting. So how we noted that point? Yes, you are there?
Yes, ma'am, I'm there, ma'am.
Yeah.
I should check with that, ma'am.
Because bone marrow is the main source for the hematopoietic stem cell. So your ideology is like from the hematopoietic stem cells, only other cells will be forming, and those cells will control the number of neutrophils or the cytokines produced. Am I right?
Yes, ma'am.
So how you can tell that point?
இதெல்லாம் தோணுது ஏன் இதுங்களுக்கு போய் பண்றோம் அப்படின்ட்டு வேஸ்ட் இதுங்களுக்குலாம் பண்றது
I don't know, ma'am. I'll check it out and tell you later, ma'am.
Okay. Okay, fine. But to my suggestion, it needs a lot of trials to undergone, because in COVID-19 itself, it's everything, it's being in a trial. Nothing has come to an end. Even the plasma cell treatment is also it's not a very good success one also. In some patients it's success, and in some it's not. So it will take some years to come to a conclusion what is the exact treatment for this COVID-19. But the ideology is very good. So I also came to know a few things about it. Check what all the corrections I told, and try to make it. Yeah, thank you.
Thank you, ma'am.
Thank you so much, ma'am, for giving suggestions for her. Ma'am, Anusha, ma'am.
Okay, and hi, Kinza.
Yes.
Hi, ma'am.
Are you able to hear me? Good morning. Okay, fine. That was a very well presented presentation. I just wanted to ask you just 2 more questions, okay? I think most of it was covered by Udayamati ma'am. So what do you think is the role of a neutrophil in your body?
It's very important, ma'am. It contributes the white blood cells, ma'am. That is if a virus is affected to our body, that is the one who comes first and battle with the virus, ma'am.
Okay, so in your slide, I read that when you're using, that is when you're transplanting, you're gonna like alter the number of neutrophils with your mesenchymal stem cells. Am I right?
Yes, ma'am.
So do you think altering the immune system, our body's natural defense system, is the right choice?
Sorry, ma'am. I can't get you, ma'am.
Do you think altering our natural defensive immune system with our stem cell is the right choice?
இந்த ரூம் ஆக்சுவலா ரூமே கிடையாது.
Are you getting my point? Are you getting my question?
Yeah,
Okay.
ma'am, I got it. It's okay, ma'am, because we are going to use our own stem cells only, no, ma'am.
Okay, but will our body be able to adapt?
Yeah, it can adapt, no, ma'am, because I'm going to take my own stem cells for this treatment, not yours, so that I get rejection or something.
Okay, right. But when you're removing your neutrophils and all, the body, the immune system will be like, it'll weaken or something might happen, right? So that's what I'm asking. Do you think it'll work out?
I think it will work out, ma'am, because so
Okay.
many people are dying because of this, no, ma'am. At least we can save 50% of the people who are dying from this virus, no, ma'am.
Okay. So actually, like in most cases, when like what you say, like for COVID, they're trying to increase your immune response, right? In most cases, like the herbal medicines and all are given to increase your immune. So when you're going against it, like removing our immune itself, that is what I'm trying to say. Okay. Go through it again once, okay? And just add that out.
Okay, ma'am. Sure, ma'am.
Okay, fine. Thank you.
Thank you, ma'am. Thank you, Kinsa, for your presentation. I hope you take the suggestions from the judges and take your idea to the next level.
Yeah,
Thanks.
thank you. Thank you.
Next. Niveta Street's team is there.
Yes, ka. Yes.
Yeah.
Yes, ka.
You're ready, right?
Yes, ka.
Yeah, good. So let us welcome the next team, Niveta Sri and Yashoda, being guided by Raj sir. Herbicides are harmful to the environment and the team here comes with the greener solution. The title is Bioherbicidal Property of Capsica Papaya Seeds, a preliminary review. Over to Niveta Sri and the team. Stage is yours,
morning.
please, Ron.
Yes, thank you, Ka. Good morning to everyone present here. Now, Yashoda will start over and present the screen.
Okay, thank you, Niveta. Can I start presenting, Ka?
Yes, Yashoda, you please. Yeah, all the best.
Thank you, Ka. Is my PPT visible?
Yes, Yashoda.
Okay, good morning to one and all present here. I am Yashoda and my friend Niveta Sri, pursuing second year of biotechnology. Thank you for having us in this idea presentation. Our idea is that bioherbicidal property of Carica papaya seeds, a preliminary review. So this is our overall abstract of our idea. So this review addresses the issue to eradicate weeds using allelopathic properties of Carica papaya seeds. So our main idea is that benzyl isothiocyanate is the major content in the bioherbicide, and it is also a major toxic content present in papaya seeds. So we can easily produce bioherbicide using it. So our overall content is introduction, idea proposal, advantages of bioherbicide, conclusion. So to start up with, weeds are the major problem of crop growth. So it results in crop losses and it ultimately results in lesser yield. As the yield decreases, economy of the country also decreases. So the best solution for this is herbicide. But as herbicide is composed of chemicals, it not only destroys weeds, it also causes side effects to our plants. So it does not satisfy the demand of customers that is healthy and natural food. So another disadvantage in herbicide is when we constantly give an herbicide to a particular weed, it gets resistant to those herbicides. So now researchers were seeking for an alternative option as a bioherbicide. So the commercially available bioherbicides are Divine, College, Biomal, etc. Now currently researchers are going how we can use the allelochemicals of many plant residues and microbial residues for production of bioherbicide. So to start up with, I would like to say why we have chosen papaya seed. In papaya fruit, pulp is an edible part. Peel we can use it as a menu. So seed is the major waste we obtain from the papaya fruit. So world is turning towards how we can use the waste product and how we can utilize it for a productively useful commercial product. So that is our main motto, to use these waste papaya seeds for the production of bioherbicide. So to know that researchers are going on to produce bioherbicide using papaya leaves, but not with papaya seeds. So it has been proven that we can inhibit the growth of following seeds, namely Helminthus annus, Diamis, and Triticum aestivum. And this idea is already available in traditional way, but our aim is to produce it in scientific way with logical proofs. So management of weeds using natural plant-derived products presents an attractive and viable means of control. So now Niveta will continue with our idea proposal.
Yes, thank you, Yashoda. Good morning to one and all present here once again. Our idea is papaya is an edible and seasonal fruit which everyone knows. Seeds of papaya has been used for many pharmacological purposes for many years. The allelopathic properties are seen in papaya seeds, which are helpful in controlling weeds. So, as it has allelopathic properties, we can't just throw the seeds in the weeds and we cannot control, as it is not an effective method. So, to do it in a proper and effective manner, we have to know about the composition of papaya seeds. The composition of papaya seeds are fatty acid, crude protein, crude fiber, papaya oil, glycotropiolin, benzyl glucosinolates, benzyl isothiocyanate, benzyl thiourea, beta-sitosterol, and enzyme myrosin. Of the constituents present, the major constituent present in the papaya seed is benzyl glucosinolate, which is a toxicant helpful in controlling the weeds. Isothiocyanates are formed when the enzyme myronase catalyzes the hydrolysis of the non-bioactive glucosinolates. After BITC formation, they need to be extracted. So the extraction process is carried out in the laboratory. Next slide. Benzyl glucosinolate is the precursor of benzyl isothiocyanate, which is found in significant quantities. Our intention is to develop a low-cost natural bioherbicidal extract against the growth of weeds. The seed of quarter ripe fruit has the highest level of BITC necessary. Next slide. To utilize the papaya seed as BITC source, an organic solvent such as ethanol is required to extract the largely water-insoluble BITC from the hydrolyzed papaya seed mixture. Next slide. From the table, it can be clearly understood that the duration of this reaction could provide valuable information on factors that optimize BITC formation. Hydrolysis time of nearly 60 minutes has the highest amount of Bg content, which is comparatively lower when the reaction is proceeded up to the duration of 180 minutes. From the graph, it can be seen that papaya leaf has 2.5% of residual Bg content, whereas the whole seed of quarter ripe fruit has 8.6% of residual Bg content. From this graph and table, it can be clearly seen that papaya seed has the more amount of benzyl glucosinolate content in it. Next slide. Next, advantages of bioherbicide. Bioherbicides are organisms which are phytopathogenic useful for biological weed control applied in similar ways to conventional herbicides. The active ingredient present in bioherbicide is living organism. Bioherbicides can easily target weeds, so we get no side effects on beneficial plants or human health, lack of residue built up in the environment, effectiveness for control of some herbicide-resistant weed biotypes. Next. So, based on the studies done, papaya is an excellent source of benzyl isothiocyanate, and so it can be concluded that papaya seeds can be utilized as effective bioherbicide. And the references are given here. Thank you for the wonderful opportunity given for us to participate in this event, and thank you for Sai sir for conducting this event, and thank you for our mentor Yuvraj sir for helping us to carry on this process. Any doubts?
Thank you, Nivedita, Sri, and Yashoda. It was a wonderful review. Let us hear from the judges.
Hi, Niveda and Yashoda.
Good morning, ma'am.
It was a good idea. Nice presentation. I have two doubts. First thing is how you are making this. I mean, you are extracting the benzyl isothiocyanate.
The seeds are first dried, ma'am. Dried, and we can make it as a powder, and the powder can be sent for the extraction process, ma'am.
So only you are extracting the benzyl isothiocyanate, or on the whole you are going to use the seed?
No, ma'am. Only benzyl isothiocyanate, ma'am. It acts as an effective
toxic on weeds, ma'am.
Okay, and now my next question is, you told in one slide, I think it's the 8th slide, you told like the quantity of the benzyl isothiocyanate present is insignificant. So how much seeds you require to create some ample quantity of this benzyl isothiocyanate?
That we didn't start to work in the laboratory, ma'am. Practically, we didn't start. This is just a idea, ma'am.
You itself told it's insignificant. So how can you able to extract? So you want such a huge quantity of papaya seed to get only a little quantity of benzyl isocyane. And again, in application or in the field, you need a large quantity to to spread or you are going to do something else. So how you justify here that this idea will work out?
Ma'am,
Yes,
it is a
ma'am.
waste, right, ma'am? So we can get we can utilize it, right, ma'am? If it is a fruit, you can say that how can you use it, but it is a waste, right, ma'am? So if we can utilize many number of ways and we can produce it, right, ma'am?
Yeah, comparing to the waste, I can fine. I can tell that it is a good, but processing that waste, it's again cost a huge thing, right? Huge amount, huge work you need. So processing of that huge waste to get a small quantity of this product, it needs lot of work driving.
Ma'am, it has 8.6% of residual content, ma'am, BG content, benzyl glucosinolate, ma'am. So, how much amount we are adding in the seeds? So we get so 8.6 content, ma'am. The extraction studies, ma'am. From the
Content is
extraction
not
studies,
so good.
we have got, ma'am.
Yeah, 8.8 percentage residual content is not so good, right? Yeah, product recovery has to be something like nearly 80% at least. Not at least more than 80%. If you are getting only 8%,
ஒவ்வொரு என் வெயிட்டையும் குறைக்கிறதுக்காக ட்ரை பண்றாங்க. அந்த
so how
விஷயம்
much work you have to put
ஏறி
in
ஏறி
to
நிக்கும் போதுதான்
extract
வெயிட் தான் ஏறும்.
only the 8% of the residue?
From one seed, we get 8.6%, ma'am. It's from single seed, ma'am.
ஹோம் ஒர்க் அவுட் சொல்லிட்டு ஆப் இருக்கு சார். செம்மையா நல்லா பண்ணி பார்த்துருக்கேன். வெயிட்
Okay,
குறையணும்னு
my suggestion is that if you are coupling with some other thing, I think it will work out.
வச்சிருக்கீங்களா?
Because only concentrating on this benzyl isocyane, if you
விஸ்கி
could
நீ
able to
பேசுறதெல்லாம் பேக்ரவுண்ட்ல கேக்குது.
identify some components present in the
எப்படி?
papaya seed, and if you could able to extract that also, it will be a good result,
எக்கோ
right?
கூட அடிக்குது.
So both together can be
ஓகே
a
அவங்க
good
கான்டென்ட்
herbicide. So this is my
Okay,
so
ma'am.
Ma'am, we'll take that solution, ma'am.
அமிர்தா
hope it will be useful for you. All the best. Thank you.
No,
Thank you, ma'am.
சார் இப்போ கேக்குதுன்றா சார்.
Thank you, ma'am. Anusha, ma'am, any questions from your
Yeah, okay. Hi, Yashoda and Niveta.
Hi,
Good morning,
good
ma'am.
morning, ma'am.
Good morning. Okay, that was a nice idea. So I just wanted to know whether your BG content that is being extracted will be based on the species of the papaya that you use, like a different location or anything, geographical specifications.
மைக் ஆஃப்ல தான் இருக்கு.
Carica papaya seeds we are using, ma'am.
Okay, will it differentiate or will it get differentiated based on where it is planted or anything?
எக்கோ இல்ல சாரோட மைக்
It is planted in higher production only, ma'am.
High production, any places specifically?
In India, we use, ma'am.
Okay.
Other places.
I'm asking because there are many types. Like for example, there are even papayas which do not have seeds. So will there be any differentiation in that? That is what I'm trying to say.
Yes,
No,
ma'am.
ma'am.
There will
Seed.
be
எப்போ உனக்கு கசிக்கிடுச்சு?
Yeah, you
because
get
it grows because it grows in different places, right, ma'am? So climatic condition also plays a major role.
Yeah.
So there will be some differentiation in BG content, ma'am. But commercially they were producing with leaves. But when compared to that, when we get a BITC from seeds, it will be much productive. productivity, ma'am.
Okay. So why do you come to a conclusion that carica papaya seeds will have better BG when compared to any other types? Like, why do you select this specific type?
Carica papaya is grown in our India, ma'am. So, which is available here, ma'am. So,
Okay, for your availability basis.
Yes, ma'am.
Okay, so my next question is how long do you think it will be effective? How long will it be effective against a weed or anything? I'm just approximately is fine because it's not a practical aspect. I'm just asking your ideas. Is there any references?
When compared to powder content, in liquid content, it will be much effective, ma'am. We are studying that only.
Duration.
I think it will be up to 2 or 3 days, ma'am. I'm not sure, ma'am.
3 days. Okay. So you have to keep reapplying it again and again.
Yes, ma'am.
Then again, where will you go for? Because as Udyamati ma'am asked, there will be the yield concentration will be very less. So how will it be effective if you're gonna reapply it again and again on an application basis?
Cost, ma'am. But we can get it in low cost. For producing it, we require only low cost. So as it reduces the cost, we can easily produce it, right, ma'am?
Okay, so, I'm just saying, for example, if you're having 100 grams of seeds, okay, and you're getting like 1 gram of your yield, okay, after drying and everything, 1 gram or 5 grams, approximately. I'm just asking, is that right or you'll get more?
No, ma'am, we get more, ma'am.
We get more, okay. You say your suggestion, how much will you get?
For 100 grams of seeds, we get up to 75 grams.
So much?
60 to 70 grams we get, ma'am.
Okay, fine. That is also okay. How will you keep applying it? You have to think about that as well, right? If you're creating something and it is effective for like say 1 year or 2 years, then that is fine. You have to apply it every 3 days means you need large quantities, right?
No, ma'am, that we are not sure, ma'am. When we practically apply and see only.
Okay, fine.
We can
just think about that as well. Go through about that as well. Sure. Okay. Thank you.
Thank you, ma'am. Thank you for your suggestion, ma'am.
Okay.
Thank you for both of your suggestions, ma'am, and thank you for giving this wonderful opportunity to present here. Thank you so much, sir.
Thank you, Niveta and the team. I hope you take this suggestion from our judges and take it in a positive way.
டேய் யூடியூப்ல போயிட்டு இருக்கு
Next
நம்ம
team.
எடுத்து போறோம் தானே இப்போதான் லைவ்ல இருக்கு
Muthusalvi's team is available. Are they available? Muthusalvi, Ayurika.
சார்
You can just unmute yourself and let me know.
லேப்டாப்
Yes, sir. We are available.
Yeah, good, fine. Let us call the next team, Muthusalvi and Ayurika Tripati, being guided by Ramya ma'am, to present a very important topic on bioelectricity. The title of their presentation is Compact Microbial Fuel Cells.
சார் அப்படின்னு சொல்லிட்டு
Over to Muthusalvi and the team. The stage is yours. Rocket.
Okay, let me share the screen.
ஜூம்ல பிரச்சனை இல்ல
Yeah, your screen is visible, Muthu.
Yeah, okay.
ஆகி போறது
A very warm welcome to everyone. And I am Muthu Chelvi from the Department of Biotechnology, VT', High-Tech, Dr. Rangarajan, Dr. Sakutala Engineering College. And we are here to present about the topic of compact microwave fuel cells in the power generation. And my teammates are Ayurika Tripathi and Meghna NVP. Is my screen visible?
Yes, Muthu, you can go ahead.
No, it is visible. Thank you. And in this presentation, we'll be seeing about the interaction of the MFCs, working, and which organism we are using, and the conclusion will be our suggestion and the references. So, interaction. And we all know that microwave fuel cells is just an bioelectrical system which converts the chemical energy to electrical energy by the action of microorganisms. These are constructed using an anode or a cathode, or you can also call it as a bioanode or a biocathode. The electrons will be produced due to the oxidation of the substrate, and the substrate can be of wastewater, saline water, or even water polluted by heavy metals like uranium and iron also. And these organisms oxidize the elements present in the substrate, and it will transfer the electrons to the electrode. And it can also transfer the electrons by its redox mechanism, and that will be carried out using their extracellular matrix. That will be forming the biofilm, and later slides we will see what is a biofilm and how it is formed. And then working. MFCs work in a very simple manner. That is, it consists of two chambers. One is an anode and another is a cathode. And these two electrodes are separated by a proton exchange membrane so that the deflections and the power drop will not occur. See, and next is that when you are using an organism like a bacteria, the bacteria will go and attach the anode and oxidize the electrons. Here I have just mentioned about the glucose, that is, it can be present in the wastewater. That which travels through the medium, and the power is generated. So depending on the application, the biocathode can be abiotic or biotic in nature. So the organism which we are going to use is a geobacter, which is a proteobacteria. Proteobacteria is nothing but a major phylum of a gram-negative bacteria, which has an outer membrane and composed of lipopolysaccharides and cytochromes. Mostly they are motile. They use flagella for their movement, and when it comes for a non-motile, they will be using a bacterial glide. And the geobacter is an organism which is found to be one such good bacteria which can reduce the uranium and the petroleum compounds and the and the geobacter is being widely used in bioremediation of oil spills like that. See, the biofilms, now we have going to see about what is a biofilm. Biofilm is nothing but the microbial secretion of an extracellular matrix surrounding the organism. The geobacter species have the ability to transfer electrons to insoluble minerals, electrodes, and these species are capable of creating thick networks of biofilms of the anode on the MFC for the extracellular electron transfer. And the first Geobacter species was Geobacter metallireducens, and it was first isolated in 1987 from the Pocomac River, Washington, D.C. They found in the contaminated ditches, okay? And these are the two pictures which species we are going to use. One is Geobacter anidoreducens and the other is Geobacter sulfureducens. They have the flagella around them, which will be, and they also have a nanofiber, and that will be very much useful in the power generation and the electron transfer. And this is how the biofilm looks. So let's go to the nanofibers. Nanowires are something which can conduct the electricity, which can be related to the neurons of in the human brain, which is able to correct the electricity. And this was first discovered by Derek Lowley and Jun Yo in the study where the memorizer experiments were taken out. And they found that these nanowires of a single bacteria can conduct electricity up to a sub-level of 1 voltage. First, in the previous cases, nanowires are considered to be the modified pili. And later on the further studies, they have been said and proved that they are composed of stacked cytochromes, namely OMSC and OMSZ. OMSZ. They come under the cytochrome type C. And these two are effectively used in the transfer of Fe3 plus ions. And you can see, right, the blue color thing, these are the nanowires, and which facilitate the long range of electron transfer across the thick biofilms. And let me say, what is a biofilm? A biofilm is nothing but a syntrophic consortium of the organism, where the organisms stick to each other and they stick to the surface also. And this is how the organism grows, by adhering to the anode surface. And this is how the biofilm look like. And the conductive pili is the thread-like structure in the picture, and the electrons will go. Am I audible?
You are not. For some time, your voice was not heard.
Yeah, I think some technical glitches.
Now it is heard, Muthu.
Thank you.
Now you can go ahead, please.
Okay, and these organisms will adhere to each of the cells and will get embedded on the anode, which form the slimy like extracellular matrix. You can see that the biofilms will be like a liquidy, slimy structure. So let me tell you about the species description. Geobacter species were considered, which we consider are Geobacter sulfureducens and Geobacter anidoreducens, and we are proposing that we are using a mixed culture. And the sulfureducens have been widely used in MFCs, And
So your voice is not heard.
now am I audible?
Yes, now you are.
I am sorry for the technical errors.
No problem. You can just go ahead and try to.
Okay, thank you. Just one more slide. I'll finish it up.
Yeah, please.
And the sulfur reducers have the ability to oxidize the organic compounds to Fe3 plus ions and the CO2 and other metal ions. And these species have given a nickname like electrogens. Another species is like Geobacter annuloreducens, which is also a gram-negative and error tolerant. Which can be error tolerant means it can live without oxygen also, and it will not get mutant or it will not stop its activity while if there is any reactive oxygen species in the substrate. So what we are proposing is that, and that will be our conclusion, we are going to use a mixed culture so that a power production will be the effective way. So the growth rate will also be in a higher rate and it will be also cost efficient. Thank you.
Thank you, Muthuselvi, for the presentation.
And these are the references. Thank you.
Udayamati ma'am, let us hear from your side. How was the presentation?
Muthuselvi.
Yes, ma'am.
Good morning.
Good morning, ma'am.
Yeah. My first question is going to be, what the innovation you are trying to tell in this?
Like already species, they are using in a single species, ma'am. And we are going to use a mixed culture and we are going to see how it's going to be in a higher product yield, or in that way we are going to present it, ma'am.
So the mixed culture is the thing you are telling, it's a different view.
Yeah, ma'am. Only a comparative study is being going on in a different cells, ma'am. And we are proposing that we can put those two organisms in a single MFC and we can see how it's going to be effective or not.
Why can't you go and trial with the other species? Why only with Geobacter?
Geobacter is it's easily available, ma'am, in the underground earth itself. On the availability basis, that's the first point. And only the basic requirements, it can grow in any condition. It can withstand the high pH, high salt conditions, etc.
Okay. So you are just trying to propose here, but there is no study evident with a mixed culture of this microbial. Okay,
yes,
so you don't know how the output will be, so you don't have any idea about it.
Yeah, ma'am. When we get a lab or something, we'll be starting with our work, ma'am.
Okay, fine. And I felt some discontinuity in your slides because beginning you were explaining, and in some I thought if you could explain that microbial fuels are working with a figure or something, because to new person who doesn't have any idea about it, by seeing the figure we can able to quickly observe it.
I have a picture, ma'am. I think maybe technical issues that I run through the picture fastly, I guess.
But it was in working you didn't show that picture. I think after that biofilms and nanowires you were showing that. I guess so. But I
Yes,
felt
ma'am.
some discontinuity in your slide preparation. I don't know why you just suddenly were explaining and you suddenly jumped to this biofilms and nanowires. There is some discontinuity in that, because while going through this, while explaining you about the microbial fuel cells, the biofilms while coming in between, it is fine, but you have taken that as a separate thing and you have explained it. Biofilms and suddenly a nanovirus is coming into play, because initially we were not discussing about this, but suddenly you are just telling all this. I felt some discontinuity in that. And overall, you are just being proposing, so I can't question you more because you are just proposing, right? So as the work progressing only, we will know what is the result or outcome coming out. So let's see, and while not for the slide preparation also, while working for your work proposal also, go with the flow.
Okay,
Don't
ma'am.
do that and don't jump somewhere. Okay.
Okay, ma'am. Thank you, ma'am.
Thank you, ma'am, for giving the suggestions to the team. Anusha, ma'am, let us hear from your side.
Okay. Hi, Muthuselvi.
Hi, ma'am.
Okay. I just have two questions. So you said you're continuing from the earlier. You said because you're choosing Geobacter because it's more available and all, right?
Yes, ma'am.
So are there any other examples other than Geobacter?
We have Svenella and other species, ma'am.
Okay. So what happened to them? Is it's not available or?
It's not that much available in India like Geobacter, ma'am.
Okay, fine. So only the availability or yield or any other reasons are there?
Like it can grow faster in high salt conditions and pH also. If there are any fluctuation in the electrolyte also, it can withstand them, ma'am. That's why we are choosing Geobacter.
Okay. So the next question is, how do you confirm that you're dealing with Geobacter?
It's a gram-negative bacteria, ma'am. We can use as staining also.
Okay. So staining is the only confirmation that you use?
No, ma'am. We can go for SEM and SEM analysis also.
Okay. So when you're going to use it, so you're saying that you have to confirm it and then only use it, right?
Yeah, ma'am.
Okay.
And some Geobacter can show positive staining also, gram-positive staining also. We need to do another study like SEM or another microscopic study to confirm that.
Okay. So you're planning to confirm it before using, right?
Yes, ma'am.
Okay. So does it have any other contamination issues and all?
No, ma'am.
No. Sure.
Sure, ma'am.
Okay, fine. Thank you.
Thank you, ma'am.
Thank you so much, judges, for your comments. Next, let us. Ananda Silence team, are they available? You can just unmute and.
Yes, we are here.
Good. Now let us call the team of Ananda Sainan and Gautam, being guided by Sarami sir. All that glitters is not gold. All rayons are not the same. The team comes out with the title Enhancement of Rayon using bacterial cellulose by Q-ammonium process. Over to Ananda Sainan and the team. The stage is yours. Rock it.
Is screen visible?
Yes. You can go ahead. All the best.
Good day to everyone present here. Myself Gautam and
myself Ananda Sainan.
We are here from second year of biotechnology. We are here to present the idea on enhancement of rayon by Q-ammonium process using bacterial cellulose. Contents of this presentation starts with introduction, followed by our idea, then comes our methodology, next the applications, literature reference, and finally our conclusion. First we see about rayon. Rayon is a regenerated cellulose fiber which is made from cellulose that we get from wood pulp, cotton, etc. It is prepared by cuprammonium process. Rayons are easily biodegradable, soft, durable, and they're highly absorbent. Rayons can further processed into fiber and they can be used for various purposes like clothing in textile industry and for making medical kits. Rayon can also be used for making carry bags, etc. Now in general, rayon was produced by cellulose that we get from cotton and wood pulp. Suppose if we use wood pulp as the raw material for rayon production, that causes to cutting down of trees which leads to deforestation. And another thing, the price of cotton was slightly higher and nowadays the demand was increasing. In order to overcome this, we thought that we can use bacterial cellulose as a raw material for the production of rayon instead of using wood pulp and cotton. Here the cellulose was produced by the bacteria. By using this, we can get more yield and the process is much easier. The rayon produced from this bacterial cellulose are soft and comfortable. As of now, for the production of rayon, cotton is used as the raw material because it contains cellulose. But cotton takes more time to dissolve in the Swedges reagent. Instead of using that, we are using cellulose produced by bacteria called Acetobacter xylenum. The bacterial cellulose can easily dissolve in Swedges reagent. Here comes our methodology. First, preparation of medium. The medium is prepared using peptone, fruit paste, and vitamins. The bacteria Acetobacter xylenum is inoculated at an optimum temperature of 28 to 30 degrees Celsius, followed by the production of cellulose by bacteria. Bacteria is added in the medium and it converts the glucose into cellulose. Fermentation takes place to produce energy for the growth medium. Then preparation of Swedges reagent. Swedges reagent is prepared by precipitating copper sulfate and then we are adding it with ammonium solution. Then the bacterial cellulose we get is added into this reagent and mixed well. Then the prepared Swedges reagent is treated with sulfuric acid for production of rayon and then it's dried for 2 to 3 days. The upcoming slides will be continued by Anand Sen.
Thank you, Gautam. Now we are going to discuss about the conversion of glucose into cellulose. For the preparation of bacterial media, we need nutrient, carbon, energy as a source. We are going to take waste rotten fruit as a carbon source. These fruits contain sucrose as a main source. And then yeast is added as a nutrient medium. Vitamins and buffers are added into the medium. The enzyme invertase is an enzyme that converts sucrose into glucose and fructose. First, glucose enters into the bacteria and metabolizes into cellulose and other byproducts. They are acetic acid and glucodonic acid, ethanol, and CO2. The acetic acid can be used for the production of dyeing purpose for the cellulose. Now we are going to see the biosynthesis of cellulose in Acetobacter xylenum. The biosynthesis of bacterial cellulose require 4 enzymatic steps as we can see here. The first is phosphorylation of glucose by glucokinase to glucose 6-phosphate. The next step is isomerization of glucose 6-phosphate to glucose 1-phosphate by phosphoglucomutase. Then the conversion of glucose 1-phosphate into uridine diphosphate glucose by UDP glucose pyrophosphorylase. Then UDP glucose is into cellulose by cellulose synthase. Then we are going to see the mechanism behind our Swedges reagent. It is prepared using 2 major components, copper sulfate and ammonia solution. The features of this reagent is that it acts as a solvent for cellulose. When we mix the cellulose, it becomes more stronger and durable. The bond formation is more stronger and it does not dissolve in water. Now we are going to see our entire process of our rayon production. First, fruit waste. The fruit waste were collected from agricultural sources and then they are being crushed into a juice, fruit-like extract. The fruit contain sucrose as the main source, and enzyme invertase is added to break into glucose. Then we are going to add yeast as a nutrient medium, and Acetobacter xylenum. It is a gram-negative bacteria. It has a capacity to convert glucose into cellulose. Fermentation. We are using fermentation for the energy purpose of bacteria to grow. Then after a few days, 3 to 4 days, we get the bacterial cellulose like this. Then copper sulfate and ammonia solution are mixed together to produce a Swedge's reagent. After the bacterial cellulose is being mixed with the Swedge's reagent, then H2SO4, then we are going to add the Swedge's reagent into the H2SO4 for strengthening the bond. Then we can see the rayon produced like this. After processing, we can get the processed rayon like this. We can see here. Then we are going to see the applications of rayon. We can make bag using this rayon, and also earphones and shirts and umbrella. These are the major applications of our rayon. These are the literature reference. Thus, I conclude thus we have produced rayon using bacterial cellulose from agricultural rodent fruits. They are easily biodegradable.
Thank you.
Thank you.
Thank you, Ananda, Simon, and Gautam. It was a very clear presentation. Let us hear from our judges. Udayamudi ma'am, can please.
Good morning.
Good morning. Good morning, ma'am.
Yeah, it was good.
Okay, ma'am.
So my question is, why you chose Acetobacter as xylem,
It
right?
is yes, ma'am. It is specifically known for converting the glucose into cellulose, ma'am. We are using bacterial cellulose in this process, so we need this bacteria to convert glucose into cellulose, ma'am.
Is there
These bacteria can be found in the soil and rodent fruits itself, ma'am.
Oh, these bacteria is available.
Yes, ma'am.
Okay, is there any other source of this type of bacteria? Or is there any other bacteria other than Acetobacter xylem?
That is a glucoacetobacter, ma'am. It is another bacteria. Acetobacter species, gene code is ER005. These bacteria can be used for this production.
Okay. So comparing to that Acetobacter, you feel it's good?
Yes, ma'am. We are using fruit base, ma'am. Majorly, this bacteria is available in the fruit itself, ma'am. So we are using this, ma'am.
Okay. And one more doubt is how we are extracting cellulose from that medium. You are told you are fermenting. So after fermentation, how we are recovering the product cellulose?
Fermentation, sucrose is converted into.
Using filtration process, we can observe.
Okay, how we are
Sucrose is separated into glucose and fructose, ma'am. By using this bacteria, glucose is converted into cellulose, ma'am.
Yeah, I got that. But how we are extracting that cellulose from that fermenter or after the fermentation process?
Cellulose will be found in the top layer of the end product, ma'am.
Okay.
You
So
can
you are
use that process.
Ma'am, like this cellulose will be extracted in the top layer of this, ma'am. Other byproducts will be.
Okay. So my question is, you are just simply going to extract the top layer, that's all, or you are going to filter?
Yes, ma'am. We can use the other byproducts also in other various purposes, ma'am.
Ma'am, the acetic acid we can use for coloring purpose of rayon, ma'am. It can use as a dye for drying. That also we can collect from these waste products.
Okay. This type of collection is possible in a small scale, but while going with a large scale, how you will be collecting?
Ma'am, using bioreactors like bioprocess engineering. We are going to make a medium like and we are going to collect, ma'am.
Okay. So how we will confirm the formed product is cellulose?
By microscopic view, ma'am, for glycosidic bond formation.
Okay. Okay, so you are planning to do this after coming to college?
Yes, ma'am.
Okay, very good. It is actually a good idea.
Thank you, ma'am.
Yeah, take it to the next level. My wishes for it is that only. So don't stick on this idea itself. Make that as a practical one.
Sure, ma'am.
Thank you.
Thank you.
Thank you so much, ma'am, for your comments. Let us hear from Anusha, ma'am.
Okay. Hi, Ananda Sainan and Gautam.
Hi. Good morning, ma'am.
Okay. Good morning. Okay. So my doubt is on an application basis, when you're going to use fruit waste, so how far do you think will you be able to use it, because it can degrade or those kind of contaminants can also occur?
Ma'am, for that purpose only we are treating with Swedges reagent, ma'am.
Okay, but you are treating it after isolating and fermenting and all, right?
Yes, ma'am.
So before that, how will you know that there are no other contaminants along with your Acetobacter xylenium?
Ma'am, for that we want to stain the bacteria, ma'am, for staining purpose, ma'am.
No, what I'm asking is, if you're going to do it in a lab, okay, you're doing it today, you can directly use your fruit waste. But what if the case you're going to use an old fruit waste, or it can be a day or older one? So how will you cope up with that?
Bacteria isn't harmful, ma'am, and we can sterilize to prevent any contamination, ma'am.
Okay. Will your sterilization kill your Acetobacter as well?
Ma'am,
no, ma'am, after sterilization only we are going to add it, ma'am.
You said that Acetobacter is present in your fruit waste.
There any contamination, we will sterilize, ma'am, because this bacteria isn't harmful anymore, because it is naturally present in the fruit, ma'am. We are to produce glucose into cellulose, ma'am.
Preparing the medium, next we can add this bacteria, ma'am, for culturing purpose.
Okay, but when Udayamati ma'am asked, you said the Acetobacter is already present in the fruit.
Ma'am, for some fruits only, ma'am, like pomegranate, guava.
Okay.
And pineapple.
So you're going to add it after sterilizing your fruit waste?
Yes, ma'am. Yes, ma'am.
Okay. Just be sure, okay? Don't get yourselves confused.
Okay.
Okay. It was a nice idea. Go along with it, and do refer it more. Like you can either use alternatives instead of fruit waste also. So go through those kind of alternatives as well. And Acetobacter also. Try finding other, try comparing with other species also as well.
Okay.
It can be either effective, or try modifying your methods also. Okay?
Sure.
Okay, ma'am.
Okay. All the best. Thank you.
Thank you, ma'am.
Thank you all.
Thank you, team Ananda Sain and Gautam. I hope you take this idea and the technique to the next level. My wishes.
Okay. A special thanks to our team, my guide, Sai Ramesh sir, who are guided. Thank you, sir.
Let us, Raja Matangi scheme, are they available?
Yes, yes.
Yeah, good. Let us call the next team, Raja Matangi and Yema Madhuri, being guided by Saramesh sir. Drought and water scarcity are the major problems in many places. The team has a solution now. The title is Gene Manipulation Using Algae to Grow More Crops Using Less Water. Over to Raja. All the... Yeah.
Thank you.
நம்ம சொல்றது அப்படியே கேட்டு செய்யற பசங்களா இருந்தா நல்லா இருக்கும். நீங்க என்ன பண்ண போறதோ தெரியல.
Is the screen visible?
Yes, you can. Go ahead.
Point,
Madhuri will be starting the presentation.
Yes, thank you, Madhuri. So, good morning to all the students and teachers here, and I welcome you all to the Biotekionz event conducted by our department. And we are here to present an idea on genetically modified crops, which uses an algae as a gene source to have a better yield using the same amount of water. The next slide is a content slide. Is the content slide which has the topics which we will be explaining in this oral presentation, which has the introduction, the research gap, the idea proposed by us, and the literature evidence which backs up our idea, and the conclusion. This slide is an introduction for our and the basis for our idea. Generally, a genetic modification or gene modification in plants is mostly done in agricultural crops, and it's a process where the desired gene is obtained from another source and is introduced into the plant, after which the gene is expressed. This modification is done using 3 types of process. The first being the transgenic modification, where the genes are obtained from another different species. And the second is the cisgenic modification, where the genes are taken from the same species or a very closely related one. And the subgenic modification, where the expression of one or more genes is either made inoperative or it's reduced. And the major advantages of a genetically modified crop is that it has a better yield than the wild crops, a better resistance to pests, diseases and environmental stresses, and an improvement in the nutrient content. The research gap is that the existing wild crops struggle to give a better yield under harsh conditions, mainly under water scarcity. And the crops which use more water are much more vulnerable than the other types of crops. If these crops which use a large amount of water for growth are genetically modified to have a higher efficiency in photosynthesis process, it will obviously result in a higher yield. Therefore, if a TSC gene from an algal species of Chlamydomonas inhati is used and introduced into the plant, it will result in a better yield from using the same amount of water, which in turn addresses the food scarcity and the limited availability of water in certain areas around the world. So the idea which we are proposing is that by using the genetic manipulation processes, the efficiency of the photosynthesis process can be increased by enhancing an enzyme, which is the BPase, which is already present in the plant, by introducing the TSCA gene from the algal species Chlamydomonas inhati. This will result in an increase of the enzyme SBPase, which is produced in the plant, and it results in increased efficiency in the photosynthesis process, thereby giving a better yield. And we have chosen Chlamydomonas inhati, this particular species, because it is a green algae and is present in a moist soil. Therefore, it has a very big eye for the sunlight. And the gene used is the TSCA gene, which is a small chloroplast RNA. And the subsequent slides says that this is a C3 cycle. And the rice, if we are taking an agriculture crop, for example, a rice or maize, and this is a C3 cycle. And you can see the cedoheptylo-7 phosphate in the center of it. When this is in abundance, the process is obviously in much more abundance, which leads to a better yield for the same amount of water used. And our idea is backed up by the evidence and literature evidence, which is said by my partner Maranji.
So here is the existing idea from which our idea was inspired. The existing idea was done using a tobacco plant, and the algae that was used here was cyanobacteria. In this experiment, they have tried and succeeded the method of simultaneous stimulation of electron transport and RUBP regeneration, which provides the stimulation for photosynthesis. Increase in the level of photosynthetic enzyme in Calvin-Benson cycle has an impact on the photosynthetic rate and the yield. In higher plants, introducing cytochrome C6 is a viable strategy to improve photosynthesis. Overexpression enzyme like ceroheptulose bisphosphatase, cyanobacterial SBPase provides us the fact that increase in the photosynthesis increases the yield of the crop. In tobacco plant, the SBPase enzyme allows overexpression. The manipulation of the tobacco gene happens in the CV cycle. Then the algal cytochrome C6 is introduced to the modified tobacco plant, and the protein from the algae and the enzyme from algae that is introduced to the plant. The simultaneous stimulation of the electron transport and RUBP regeneration, SVNC6, takes place simultaneously, which results in the increase of photosynthesis and increases the biomass accumulation, which eventually results in the higher yield of the crop. This experiment was done twice. First, it was done in 2016 and then in 2017, 2 times under glasshouse condition and in normal field condition. Tobacco that was already done is a C3 plant, and our example or the experiment idea proposed is a rice, which is as well a C3 plant. So that gives us more hope that our idea of cultivating rice crop in the place of tobacco using Chlamydomonas reynoldi has a chance of succeeding. This was the paper that was published on August 10, 2020 by Nature Plants on the topic stimulating photosynthesis process increases the productivity and water use efficiency in the field, which gives us more clear explanation on how the experiment was conducted. So we would like to conclude our presentation by saying that using the simultaneous stimulation method, we can increase the productivity of food crops and at the same time not use extra water than the usual amount that the crop would require. And thereby it is beneficial for humankind both ways. We get more food crops and no extra water is being used. And these are the references. Thank you.
Thank you, Madhuri. It was a nice presentation and you have supportive joined your group. Let us hear from Udayamari.
Rajamat Hema Madhuri.
Good morning.
My first thing is nowadays you would have heard that there is so much against these genetically modified crops. Am I right?
Yes, ma'am. Genetically modified crops are safe also.
So how in this work how you are justifying that it is safe?
Sorry, ma'am. Can you repeat your question?
In this case, how you are justifying it is safe, though I agree that it is reducing the water content level, but you are basic thing you are modifying the crop. So how you are justifying it will be safe?
It is safe because there is already a done in a tobacco plant, so that says that the tobacco has a better yield without any of the consequences of not being bad. So therefore, we can be safe that it is safe for us now.
Yeah, okay. So tobacco we are not consuming, but you are stating that we are consuming the rice. We are going to consume the rice. Am I right? So in how far that rice won't get affected in this?
It's because the tobacco and the the tobacco and the rice plants are C3 plants. So they are of the same type of plant. Therefore, the backdrop will be less now. But it can only be confirmed after we do it on rice plant in a real procedure.
Okay. So my what I got from this, you are taking algae as a source for the gene production, and you are introducing the gene to the rice crop.
Yes, ma'am.
In the initial stage itself.
Yes, ma'am.
this as a seed. So you are going to inject or you are going to apply?
With inject, ma'am, it's much more efficient than applying.
Okay. So is there are so many types of rice available? Is there any particular type of rice you are targeting?
We are trying the Asian rice, ma'am, Risa sativa.
Risa sativa.
Okay. So why this algae? Because it is contained enzyme that pseudoheptulose, something it was.
Yes, ma'am. And it's also because it's a green algae, so it's much more
that is present in the soil, ma'am. It has a big eye spot that can actually take in more sunlight, so it will help in the photosynthesis.
So by comparing with tobacco and only you are suggesting this as an idea.
Tobacco, they've used cyanobacteria, ma'am. So we are using Chlamydomonas.
Okay. But comparing with the idea I'm asking, comparing with the tobacco only, you are suggesting this.
Yes, ma'am.
Okay. So how for in experimental level, how you are planning to do this?
It's still in the early stages, ma'am, because it has not been done in any of the commercial crops. It's only done in a commercial tobacco plant, which we don't use for the consumption like we do for rice. So it has first to be done in a basic level, ma'am. Then only it can be taken into a much more advanced level.
Okay. So no, I'm just asking how we are planning to do in the laboratory scale.
For now, we are yet to have a solid plan on that, ma'am. Because this is a new thing, we are yet to have a concrete plan on how to do it, ma'am.
Okay, all the best. I hope you will take this as you will take this into the next level.
Thank you.
Thank you.
Thank you.
Thank you, ma'am. Anusha, ma'am.
Okay. Hi, Rajamanthanki and Hema Mudri.
Good morning, ma'am.
Good morning. Okay. So my first question is, you compared it with your tobacco plants, right? So does your that is whoever has done the project, did they find any side effects or adverse effects while using tobacco?
No, ma'am. No side effects were shown, ma'am.
You're 100% sure that only the crop yield was there?
Yes, ma'am.
Okay. So why the specific algae you have chosen? And why algae? You could have manipulated with any other plant, right? So why specifically algae?
Ma'am, it's because the algae, this particular algae has been worked on by many other scientists for various purposes. So we know the properties of it much more than another fungi or another algae, ma'am.
Okay, so you didn't want to discover a new type or anything?
It would not.
You could have paid a new way.
It would have been more difficult and more time consuming, ma'am. It is possible to use another algae, but this algae has been worked upon and the properties are known. And it also has the required properties which we want. So this
algae also grows fast. I mean, it just takes 5 hours in the soil. So comparatively, this is more efficient.
Okay, fine. So the tobacco plant has been grown in field itself, right? Or in a laboratory setup?
They've done both ways, ma'am. They have done, in the field they have grown separately and tested it, and in a glass house they have separately. Right.
Okay, was there any difference between both of it? Or?
No, ma'am, both actually resulted in the more yield, high yield. In 2017, one of the experiments conducted did in the glass house, it did not give more yield. It just remained the same. But the 2016 experiment and the 2017 they conducted 2 experiments, and the last one gave more yield. The final experiment high yield in both glass house as well as the field atmosphere.
Okay. So the next one is, do you feel like an external factor can affect a plant other than your gene? External factors like you're gonna add pesticides or whatever on an application basis. I'm asking.
We don't think so, ma'am, because it is only, this gene manipulation is done only in the area which is in relation to the photosynthesis process. So
Okay.
the plant's natural defense system against the pesticides will be present because we are not touching that area. So as per our theoretical conclusions, there won't be any changes against the pesticides, ma'am.
Okay, fine. So on an application scale, when you're going to grow it on a like farmland or anywhere, will it be dependent upon any specific soil or climatic conditions?
No, ma'am. I don't think it will be, ma'am, because again, the natural properties will be retained within the plant with an extra property of this photosynthesis efficiency.
Okay. So you're sure that this gene manipulation will not affect the plant in any other way?
Yes, ma'am. It will only give a better advantage of the efficiency of the process.
Okay. All right. All the best, guys. Thank you.
Thank you so much, ma'am.
Thank you, Ansha ma'am, for your suggestions. And I thank Matangi and Madhuri and my wishes to take up this idea to the experimental level in the future. Thanks again.
Thank you.
Vinta's team, are they available? Yamalata and Vinta. Can you take up the presentation now? Vinda, Yamalata?
அதுக்கு முன்னாடி என்னன்னா இவங்க சாப்பிட்டுட்டு வச்சுட்டு போயிடுவாரு எனக்கு தட்டுல எல்லாமே இருக்கும் அந்த தட்டு இருக்கும்.
Okay. Jayata, are you there? Can you take up?
Yes, Ka. Thank you.
Okay, let us welcome Jayata, another single girl team being guided by Sarameshwar. The another green technology, the control of pests by insects. The title of our presentation is Pheromones and its application. Over to Jayata. All the best, Jayata. Do well.
Thank you, Ka.
அவங்க என்ன பண்ணாங்க சார் சாப்பிட்டுட்டாங்க நீ வாடா வந்து பேசுறாங்க இல்லை சார் நான் சாப்பிட மாட்டேன் வச்சிருப்பாரு நான் வச்சிருக்கேன் நான் சாப்பிட்டுப்பேன் அவர் சாப்பிட்டு அப்புறமா வச்சு அவர் சாப்பிட்டு
Can you?
Yeah, your sharing is visible. Yeah, you can go ahead.
Hello, everyone. This is Chaitra, and here to present pheromones and its applications. Everyone knows about hormones, which is secreted inside our body, to maintain the chemical metabolism of our body. So here are the pheromones, which is secreted outside our body to communicate with other organisms of same species. So these chemicals secreted outside and alter the behavior or psychology of other organisms.
Sorry for the interruption. You can just make your slide in the full screen panel. You can rotate it so that it can cover your.
Okay, ma'am.
Yeah.
Okay. So these pheromones are also.
Can you rotate on the right hand side clockwise so that you can?
Can you see it?
Left hand side, can you make it? It is upside. Yeah.
Hello. Can you see it, Ka?
Yeah, you can go ahead.
Okay. So pheromones are also called behavior altering agents. As it is produced secreted outside the body, it is also called ectohormone. These pheromones are categorized into many types, distinguished into many types according to the function they behave. Some of these are alarm pheromones, puttrial pheromones, sex pheromones, trial pheromones, etc. Alarm pheromones indicate alarm pheromones indicate that there is danger in some zone and indicate the other species to stay alert. So likewise, sex pheromones are produced to attract the opposite gender of the species. Pheromones are produced by every organism from unicellular to multicellular organisms. There are two types of, pheromones are categorized into two types according to the behavior it alter in the receiver. These releaser pheromones affect the receiver at particular time quickly, and the primer pheromone affect the psychology of the receiver for the long time effect. So, as there are so many organisms, every organism produce pheromones. Here are some of the examples I have given. Insects pheromones, particularly insects produce pheromones to communicate with each other. These pheromones are mostly hydrocarbon of volatile chemical substances, which is therefore easily received by the particular species. So these pheromone molecules get observed at the antenna of the insect, and get inside the particular species and binds with the compound of pheromone binding protein in the sensory membrane and interact with the specific receptor protein, and in which the chemical signal of pheromone is converted into electrical signal, and it is so that the communication is carried out in insects. For example, for a good example of insects, here is the everyone have wondered in the childhood that ants may go in a straight line, and as everyone is, many of the people would have known this, but ants, the first ant produce a pheromone and pheromone throughout its path, and which is followed by the following ant, so that it goes in the straight line. There are more than 400 chemoreceptors in ant itself, and 183 chemoreceptor genes in animal. Here I have given the picture in which two insects communicate with the following hydrocarbon chemical of pheromones. Even microorganisms produce pheromones to communicate with other species. Especially bacteria produce pheromones for various responses, including competence, correlation, plasma transfer, etc. Competence is ability of the bacteria to alter its genetic material. So, and sporulation is production of spores in unfavorable conditions. Plasma transfer between the two bacteria cells helps in genetic variation. There is some microorganism which produce pheromone to kill the other microorganism of same species. For example, in our human duck, there is a bacteria called Heterococcus which produce pheromone COD1 that kills Heterococcus faecalis bacteria, which is the same family of Heterococcus. So the Heterococcus, the non-resistant bacteria of Heterococcus produce pheromones that kill resistant bacteria of the same species. So So it shows that the native healthy non-resistant bacteria might prove useful in fighting the resistant bacteria infections. Okay, so now we will move on to mammals. Likewise in insects and microorganisms, mammals also produce various types of pheromones, uh, even including our human itself. So these pheromones are also present in urine, their food pads, semen, vaginal secretions, breast milk, saliva, and breath. Mostly, uh, let me explain this with an simple example in dogs. We have noticed that the dogs of one street may not enter to the other street. If it enters, other dogs could have bark and make fight with each other. So it is due to the territorial pheromones there is at the perimeter of their streets. So that the other dogs of other streets should not enter the perimeter of that dog street. If it enters, the other dogs will shout bark. So here I have given a picture of one goat get attracted with the other goat of opposite gender. So it produces some pheromone hydrocarbon, which is primary pheromone, which I already told that it creates the psychological effects of the receiver. So as this pheromone reaches the olfactory receptor, it activates the GnRH generator, which means gonadotrophic releasing pheromone, which is helpful in synthesizing the follicle stimulating hormones, and thus it enhance the reproductive neuroendocrine system. So these pheromones, as in insects, as the pheromones are accepted in the antenna, here the pheromones are accepted in vameronasal organ of the mammals. These vameronasal organ directs these pheromones and connects and sends these electrical signals to the brain, hypothalamus of the brain, which further deals with the hormonal balance in our body to respond the particular species, responses in the particular species. Okay, even plants could communicate through these pheromones. Okay, these pheromones actually some plants like willow, poplar, sugar maple release pheromone when they are attacked by any insects. So these pheromones reaches the neighbor plant and they detect that and they protect themselves by producing the anti-herbivore phenols. Or else it will also, some plants also reduce their nutritional value to protect themselves from the insects. So insects could also communicate through these pheromones. Some pheromones also produce tannins against the insects or some other herbivores. So plants pheromones could travel 50 to 100 centimeters. Some plants can also communicate through electrical voltages and system of voltages, electrical pulses and system of voltages. The voltage signals in the tissue radiated out from the site of damage toward the stem or beyond. Genes involved in transmitting the electrical signals produce channels in the membrane just inside the plant cell membrane. The channel maintains electrical potential by regulating the passage of charged ions. Okay, from this ideas of pheromones in different organisms, we could manipulate these pheromones according to our needs. And here are some of the applications which is already available in worldwide. Pheromone trap. So, as I already told, the insect pheromone plays a major role in pheromones. So, the application of pheromone trap is used to control the population of the particular insect in crop field. as we have already we have seen the usage of chemical pesticides in agriculture, which is toxic and damages our health. So here are some methods which is non-toxic and very safe for human consumption. So if the pheromones of particular sex of the pheromones of particular insect of one sex or may is prepared biosynthesis in the laboratory and which is sold in the market. So in a particular sticky surface, these pheromone traps are kept, which is attracted by the opposite sex of that these insects, and these insects get sticks on the sticky surfaces, and so that the reproduction of these insects gets stopped, and the control of the population is so that we can control the population of these insects. Okay, some animals like mice and bumblebee avoid inbreeding by pheromones. They release pheromones which is detected by the receiver of some other receiver of the mice and bumblebee, and it detects that it detects whether it is from its closely related organism or more distinct organism, so that it could inbreed with breed with the distinct organism by avoiding the inbreeding. These inbreeding can cause lower survival rates in the species. So to avoid inbreeding, these organisms produce pheromones. So we can also, in case of controlling the population, we can also introduce pheromones from which it can avoid inbreeding. So we can also destroy the pheromone which causes inbreeding, and it gets treated with inbreeding, so that the population gets reduced. Okay, when it comes to humans, it is always said we hadn't sensed any pheromones. We hadn't sensed any pheromones because the vamerosal organ present in our nose is actually non-functional. The genes code for this vamerosal organ is called podogene, which is non-functional. So we couldn't sense any pheromones produced by us. The most of the human pheromones are produced in urine and sweat. So it is some scientists also say that some of the olfactory sensors pick up the pheromones, and it was not detected by our central nervous system. So I mentioned here is as putative human pheromones because we are not sure that whether our humans could produce pheromones. So the male produce pheromones called androstenedione, and female produce estratestosterone. So some pheromone companies, some perfume companies uses the technology and produce synthetic form of these putative human pheromones and uses in perfume to attract the opposite gender. So hereby I conclude that pheromones is the non-toxic and good technology and safe technology for the control of population in particular species in which we are interested. So if we want to decrease the population of particular species or insects, we could use the pheromone, or if we want to increase the population of some rare species which we want to preserve, we can also introduce the pheromones and edit the genes which produce these pheromones. So that's why I conclude my presentation, and here are some websites I referred for this presentation. Thank you.
Thank you, Jayatha. It was a nice presentation from your side. By the way, another new judge in our panel. I welcome Ramya ma'am. Ramya ma'am, from your side, you can ask.
Thank you, Hira.
Yes, ma'am.
Yeah, Jayadra. Good morning. Good afternoon. Hello. Can you hear me?
Ma'am, it was so noisy in your side.
Yeah, you can hear me now? Hello? Hello?
Yes, ma'am, you're audible, ma'am.
Jayadra, can you hear me?
Yes, ma'am.
Yeah, okay. One second. Yeah, now it's okay?
Yes, ma'am.
Okay, Jayadra, you have done a very good study. It's very nice.
Thank you, ma'am.
Okay, so can we isolate this pheromones? It is possible?
Ma'am, we could, but the isolation may cause harm to the particular organism. So it is better to biosynthesize these pheromones in laboratory. Many steps of reactions.
So how you can biosynthesize it?
Ma'am, actually I studied, but I think it is so upgraded. I couldn't understand those. Sorry, ma'am.
So you told like to stop the population, we can stop the production of pheromones, right?
Yes, ma'am.
So I need to stop the population of one kind of species. Okay.
Yes, ma'am.
So at that time I can isolate this and I can give it to another organism, or I need to change the gene of another organism or another species.
No, ma'am. For example, I have mentioned the pheromone trap in which the biosynthesized form of pheromone is kept in the sticky surface so that the insects get attracted to the pheromone and get attached in the sticky surface and cannot move. So the one gender of that species gets trapped in the surface so that the reproduction gets stopped and in that area the insect population will get reduced.
Okay. So to increase the population, what we have to do?
To increase the population, I think so. I have mentioned the perfumes in human pheromones. They introduce pheromones in. You can simply make the biosynthesis of the pheromone and just spray it on the insects of the insects in the opposite genders. Or we could manipulate the genes of the pheromone so that it produces more pheromone to attract its opposite gender.
Okay, so you are saying that we can modify the species gene to produce pheromones.
Yes, ma'am.
Okay. Or we have to biosynthesis it and we have to give it to the particular species.
Yes, ma'am.
Okay, so if this work has been done before or any studies there?
No, ma'am. I don't know about the spraying method, but I know about that gene method. Some species have get modified by to produce less pheromones or more pheromones.
Okay, so it has been done already.
Yes, ma'am.
Okay, but the perfume like spraying idea, you are giving the idea or it is also proposed?
No, it is my idea.
Okay, you are saying that you can biosynthesis it and we can spray it on the on the species.
Yes, ma'am.
So how can you spray it on, like you are saying that it produces a sticky layer. So if you spray it on an organism, if you spray it on a species, the other species will come and stick on this particular species.
Yes, ma'am. Yes, ma'am.
Okay. But we can't do it in all the species, right? So in humans, we can't just like that spray the.
Yes, ma'am. We can use. That's why we are producing the perfumes. The perfume companies are already using the pheromone techniques to attract the opposite genders.
Okay, fine. Okay, thank you.
Thank you, Ramya ma'am.
Thank you.
Ma'am, let us hear from your side. Any questions, ma'am?
Hi, Chaitra. Good morning.
Good morning, ma'am.
Yeah, actually, what you are trying to propose here, I could get those 2 points. Like you can able to control a species, or you can able to make the species multiply if it is end species.
Ma'am, we cannot.
Sorry, extinction state, you can able to multiply that particular organism. And if you want to control that organism, we can make it to control. So this is the point you are trying to.
Ah, yes, ma'am.
These 2 points. Okay. So, but why you take the first point in level? Why you
Ma'am,
make how you can
couldn't hear your, ma'am, I couldn't hear your voice properly, ma'am.
Now you can hear.
Ah, okay, ma'am. It's
So why you didn't elaborate that first point? Biosynthesizing pheromones externally to control the other species.
because I couldn't understand the journal of the biosynthesis fully, ma'am. So I don't want to explain it.
By making that so only this entire idea will get completed, right? It's
Ma'am,
like
I didn't say, actually it is upgraded, ma'am. Those process and reactions are upgraded. So I want to learn.
Okay. Okay. All the best. Concentrate more on this and work on
okay,
it.
ma'am. Thank you, ma'am.
Jaitra,
ma'am.
It is produced by microorganisms also. Pheromones.
Yes, ma'am.
Okay, the property is same. Pheromones have only single property, or it has some other properties.
It is produced in microorganisms. It is produced for the response of competence, correlation, plasmid transfer, and bioluminescence also. So in microorganism itself, they could communicate with each other using these pheromones, especially in bacteria.
Okay, so it's only used for communication purpose.
Yes, ma'am.
Okay, see whether if it has any other functions and can it be isolated from microorganisms. Like you can, like for laboratory work, you can see whether it can be isolated from any microorganism. And after isolating the pheromones, whether it can be used for any other applications.
Yes, ma'am. I have seen that, ma'am. But in yeast organism, they have isolated the pheromone, which they have worked against the insect pheromone to control the population.
Okay. Okay, ma'am. Thank you.
Thank you, ma'am.
Anusha, ma'am, from your side, any clarifications, ma'am?
Hi, Jaitra. Good noon.
Good morning, ma'am.
Good noon.
Okay.
Okay. So you said the insects or microorganisms, they produce these pheromones, right? So have you gone through the point that what happens to an insect when it produces these pheromones, or what initiates these pheromones to be detected by another insect?
Ma'am, the structure of the antenna in the insect, which the pheromones get observed in the structure of antenna, surface of the antenna, through which they will, so in micropores in the antenna, the pheromones get inside, pheromone molecules get inside the micropores of the antenna and gets to the syncytium lymph, which is called syncytium lymph, which is lyophilic, and the pheromones are the syncytial lymph are hydrophilic and the pheromones are lyophilic, so that it get inside without so it fits inside it smoothly and get and binds with the pheromone binding protein, and the receptors in the
chemosensory membranes and the receptors in chemosensory the receptors in chemosensory membranes transmit the electrical signal of pheromones to the electrical signal through the central nervous system.
Okay, so these move around as signals.
Ma'am?
They move around as signals to the central nervous system. Right?
Yes, ma'am.
Okay. So I just wanted to clarify another thing. In your slide, you mentioned human consumption. Can you go through your slide once?
Human consumption.
Yeah, they are non-toxic and they can be consumed by humans. I did not get what you were trying to say there.
It was non-toxic and it does not like the crops produced by the crops produced from the pesticides or have health
crops from
health.
pesticides.
Yes, ma'am. Crops from pesticides have health issues, but these technique from pheromone
okay,
does not give any health issues and safety use for the human.
Okay, so on basis of agricultural production, you're trying to say?
Yes, ma'am.
Okay, so what does it does to a plant?
Actually, it does not do any reaction to the plant because these pheromones are kept in the sticky surface of the particular area. In the only the insects get affected by these pheromones. So the plants do not get affected by these.
Okay, so you can you can replace pesticides by using these pheromones. That is what your point is.
Yes, ma'am.
Okay, okay, fine. The ideas was good, but just go through the how it is occurring and all. Just go through it a bit more, okay?
Okay.
Okay, thank you.
Thank you.
Thank you all the judges for your suggestions for Jayata.
Thank you.
Yeah. All the best for your work. Next team, let us welcome Jai Vignesh. Jai Vignesh team is available.
Yes,
are you?
Yes. We are here.
Let us welcome the team Jai Vignesh and Mukundan, being guided by Ramya ma'am. Biofertilizers are environment friendly and nitrogen fixers form an important group. The team comes with one such species. The application is isolation and enrichment of Acinetobacter. Here they go. Over to Jai Vignesh and the team.
Thank you.
All the best.
Thank you.
Our screen is visible?
Yes, you can go ahead.
Okay.
Yes, sir. Good afternoon, one and all present here. So I'd like to start with our explanation. So we are going to speak about the, actually our final result is to produce a biofertilizer. First we are going to isolate Acinetobacter species, and with the help we are going to make that. The topic is isolation and enrichment of nitrogen fixing bacteria Acinetobacter species. Okay, myself Jai Vignesh, second year biotechnology.
And myself Mukundan, second year of Department of Biotechnology. Then next we going to the contents. In contents we have introduction, idea proposal, methodology, application, literature and reference. And last conclusion.
First slide.
Introduction. In this introduction, Acinetobacter belongs to the family of Pseudomonaceae and Acetobacteriaceae and class Proteobacteria, which is common in soil samples from across the world. They are aerobic free-living soils microbes that plays an important role in the nitrogen cycle in nature, binding the atmospheric nitrogen, which is inaccessible to plants, and release it in the form of ammonium ions into the soil. It's a nitrogen fixation. By the ash-based mannitol's broth or agar are used for the cultivation of the Acinetobacter species that can use mannitol and atmospheric nitrogen as a source of carbon and nitrogen respectively. We propose that the idea that production of biofertilizer from Acinetobacter species. We are going to isolate and enrich the
Azotobacter species from soil samples using ash-based mantle broth and use the bacteria to produce the biofertilizer. There are many other broths with the help of which we isolate the nitrogen-fixing bacteria species. But the purpose of the using ash-based broth is to produce a large colonies of Azotobacter species with efficiency and quality. The required ingredients to make the broth. We calculate that the ingredients according to grams per liter. The mannitol is 20 grams per liter, dipotassium phosphate, magnesium sulfate, sodium chloride, potassium sulfate, calcium carbonate, and a final pH value at 25 degrees Celsius within the 7.4 plus or minus 0.2 at the rate of pH. And the agar at 3.5% is added to the above composition. We get the ash-based mannitol in the agar medium. Procedure was continued by J. Vignesh.
Thank you, Mugundan. So we'll see the procedure for making ash by mannitol broth. First, we need to weigh all the ingredients one by one using balance. Then we need to add all the ingredients one by one: mannitol, magnesium sulfate, dipotassium phosphate, sodium chloride, and potassium sulfate. Then adding 100 ml of distilled water to the flask, and we need to dissolve all the ingredients. pH should lie between 7 and 8 so that it will be useful for which is required for the growth of microorganism. Then we need to add calcium carbonate to the medium so that it gets thickened. CaCO3 is insoluble, so we need to dissolve the medium by shaking thoroughly so that it gets evenly distributed. Then we need to sterilize. Now we have prepared the ash-based broth. This is what ash-based broth, and we need to sterilize the broth by autoclaving. The autoclave should be maintained at a pressure of 15 psi and for 20 minutes, and we need to monitor the pressure. Then we need to sterilize. After sterilizing, we have sterilized the broth. Then we need to add a gram of soil to it. We need to inoculate the soil. Then incubate it for 2 days at 30 degrees Celsius. Then the speed of rotation should be 120 RPM. Then we need to, after incubating, the Acetobacter species in the broth is enriched, so we can isolate now. Now the isolation process is carried out by quadrant streak plate technique. We need to incubate the plate at 30 degrees Celsius in an incubator for 3 to 4 days, and after incubation, we get colonies of nitrogen-fixing bacteria on the agar plate. Then we have finally isolated. For the production of biofertilizer, we need to make a dense suspension of purified culture in sterile saline. Then after saline suspension of purified isolate. Then we can add charcoal powder as a medium for usage as a biofertilizer. The proportion should be 1 ml of culture per gram of charcoal powder. Then we need to shake the flask well and mix the culture well with the charcoal powder. So after purifying it, we'll be ready with biofertilizer. Then application of biofertilizer are many and numerous. We know that Acetobacter improves seed germination and has beneficial response on crop growth rate. It helps to increase nutrient availability and restore soil fertility for better crop response. It is an important component of integrated nutrient management due to its significant role in soil sustainability. Acetobacter species especially are freely available, free-living, non-symbiotic heterotrophic bacteria, and they are capable of fixing an average of 20 kg nitrogen per hectare for a duration of per year. And these bacteria are regarded as plant growth promoting rhizobacteria, which synthesize growth substance that enhances plant growth and development and inhibit phytopathogenic growth by secreting inhibitors. It also helps in nutrient uptake and some other and produces biochemical substances such as proteins and amino acids. Then finally, there are literature reference here. We refer these references. Then finally, we conclude that we have isolated successfully the Acetobacter species from ash-based mannitol broth. We can use this fertilizer for plants like paddy, wheat, and sugarcane, which are often which often require chemical fertilizer, and they cause plant diseases and other disadvantages. So we can replace it with this biofertilizer, and it requires nourishment in shorter duration. So they are treated with chemical fertilizer. So in order to overcome that adverse effects, we can use this fertilizer. There are many adverse effects of using chemical fertilizers like water pollution, chemical burn to the crops, acidification, and mineral depletion of the soil. The best way to overcome is the usage of biofertilizer. Then the isolate can be made as biofertilizer. That's the end of the slide. Thank you. Any query?
Thank you, Jay Vignesh and Mukundan.
Thank you, sister.
Thank you.
Judges. Udayamadi ma'am.
Yeah, hi, Vignesh and Mukundan.
Good afternoon, ma'am.
So, what is the novelty you're trying to tell in this study?
The novelty or the innovative is that we are using this particular type of broth to increase the number of bacterial colonies, ma'am. That Acetobacter species colonies are increasing in the agar plate, so that we can increase the efficiency of biofertilizer.
But this ash-based mannitol, you are telling, no?
Yes, ma'am.
But it's already commercially available.
Yes, ma'am.
Yeah, so what is the innovation in your thing?
Yes, ma'am. There are many other broths, ma'am, but we are particularly using this for the increase in productivity and.
No, that's what. There are many other broth, and this particular broth is actually commercially available. Ash-based mannitol agar is commercially available. So,
but we are mixing it. We are mixing it with charcoal, ma'am. But I think that is not already available. Then we can also add other ingredients like seaweed and other nutrient compounds, so that it will get differ and we can improve the plant growth and production.
so you're going to add the thing which reduce the weed production also.
Yes, ma'am. Then our other idea is to co-culture other species of bacteria, ma'am. There is not only Acetobacter, there is also other species like Rhizobium and Azospirillum species, ma'am. They can also fix the nitrogen from the atmosphere, and they can also be used for this purpose. We can do the same procedure for those kind of bacteria also, ma'am. And we can use it as biofertilizer.
Okay. So, but let Anusha ma'am, you can join.
Yeah, ma'am.
I will add some more notes up.
Okay. Okay, so hello.
Good afternoon.
Jay Vignesh and Mugundan. Good afternoon. So, can you tell me why did you use ash-based mannitol agar?
Yes,
the specific use.
Specific use is this ash-based mannitol agar enhances that particular Acetobacter species, ma'am. It provides all the nutrients that is especially suited for that particular bacteria. So
okay, can you give me certain examples of the composition that is necessary or the reason that it enriches the bacteria?
A reason that it enriches, because it is already Acetobacter is naturally available in soil, ma'am, across the world. It is naturally available. So we need to enrich it with the specific nutrients so that it is available in that broth.
Okay, that's what I'm trying to ask. Which is the nutrient that is essential for enriching this Acetobacter from your media?
Yes, ma'am. We have listed
which
that.
one?
Mugundan, can you share that slide? Yes, ma'am. Ingredients, yes, ma'am. So the ingredients
I'm asking which is the specific ingredient that is enriching it, because certain chemicals will be present in almost all the media. So what is your
specific ingredient? Yes, ma'am. Specific ingredient is mannitol especially, and other ingredient is calcium carbonate, ma'am. The other ingredients are generally available, general, and these two ingredients are especially required for the
Okay.
nutrient.
Okay. So are there any other alternate medias which can be used instead of ash peas?
No, ma'am, we are not sure of that, but we can use alternate bacterial species, ma'am. I already mentioned that we can use Rhizobium and Azospirillum species also, ma'am.
Okay, that is fine. I'm just asking whether you like went through the varieties of like agas that is already available for your process. Okay.
Yes, ma'am.
Don't come to a conclusion that ash peas is only that can
Yes,
be
ma'am.
used. So
Yes,
that's what I'm trying to say.
yes, ma'am. Yes, there are also other broths which can also be used.
Okay, all right. So why did you use charcoal as an enhancer? You use charcoal, right?
Yes, ma'am. We need to finalize it in the form of biofertilizer, ma'am. So that we need a material which is available cheaply, so that we use that charcoal, ma'am.
Okay, so what about rice husk or any other grains?
Yes, ma'am. But we need to make this practical and make sure that it's working. We need to check with
No.
the other
So
companies.
what will your charcoal do to your biofertilizer that is already prepared? What is the role of charcoal?
Charcoal also in it is also acts as a nourishment compound to the soil, ma'am. So it does not affect our. We need a medium for our biofertilizer, ma'am. That's the purpose of using charcoal.
Okay.
So it does not affect our nutrient availability.
Okay. Will your charcoal affect your acetobacter?
No, ma'am. No, ma'am. It doesn't affect any of our components or nutrient availability.
Okay. So what I'm trying to say is, you said that you're going to add this property and you're going to add rhizobium, acetobacter, then charcoal, all that, right?
Yes, ma'am.
So when you're going to add a lot of composition to your biofertilizer, make sure it does not affect the initial idea of what you're trying to do. Okay?
Okay, ma'am.
Okay. If you're targeting on acetobacter alone, then make sure that it is standing out in your biofertilizer. Okay?
Okay, ma'am.
Okay. When you're combining a lot of things, you have to make sure that each component is doing its job.
Okay, ma'am. Okay, I understand.
Okay. So go through it a bit more and come up with a solution. All right?
Yes, ma'am. Thank you.
Okay. All the best. Thank you.
Thank you, ma'am.
Yeah, I will add some more things. Since this medium I was telling you, it is already commercially available. So your thing, what you are trying to say is the charcoal, what you are adding is a major innovative out of this work. Am I right?
Yes, ma'am.
Okay, so why can't you try to build some more things to add up in this?
Yes, ma'am. We need to work on that, ma'am. We can also add other nutrient supplements, other waste materials which can be added, but we need to think of that.
Think of the organisms also.
Yes, ma'am.
Not only focus on Astobacter, what other organisms can also be used?
Yes, ma'am. In this procedure, we mentioned that we are using a gram of soil sample in the broth, so that Acetobacter is available easily in the soil, ma'am. So that we use that.
Okay.
The purpose of making using that is it's the only reason, ma'am.
Okay. So come out with a well-versed idea and make it practical. All the best. Thank you.
Thank you, ma'am.
Thank you, judges. Vrinda's team, are you available? Or we can. So, Sai sir, they are not available, I think. Shall we?
Yes, they are not available. Close
இரு ஒரு நிமிஷம் சிவா ஒரு நிமிஷம் நான்
thank you.
ஹீரா
Yes, sir. Shall I close it?
yeah
End up.
let me just thank both the evaluators and then we'll close it. We'll have to close it.
Thanks, sir, or you can.
Yeah, I'm doing. Udayamati ma'am.
Okay.
Yes, sir.
Thank you ma'am. Personally thanks because you took lot of time in explaining
It's
things.
my
In a serious the helpful irundhathu. I was expecting such contribution from you. Serious nalla irundhathu ma'am. Thank you. Personally thanks. And
Yeah, it's my pleasure.
thank you ma'am. Anusha ma'am.
Yes, sir.
நீங்களும் ரொம்பவே ஹெல்ப் பண்ணீங்க மேம் தேங்க்யூ. ஏன்னா ஈவெண்ட் கண்டக்ட் பண்ணதே அதுக்கு தான். பீப்புள் வாண்டட் டு லேர்ன். ஏன்னா இவங்க இப்போ தான் ஸ்டார்ட் பண்ணுறாங்க. ஸோ இட் வில் பி ரியலி ஹெல்ப்ஃபுல் ஃபார் தெம் யுவர் சஜஷன்ஸ். ஐ ஹோப் தே டேக் அப் தி சஜஷன் அண்ட் சேஞ்ச் த வே தே ஆர் ப்ரெசென்டிங் ரைட் நவ் டு அ பெட்டர் ஃபார்ம். ஸோ தேங்க்யூ போத் ஆஃப் யூ. தேங்க்யூ மேம்.
Our pleasure, sir.
Hira, you can close the meet, Hira. Thank you, participants.
Yes, sir.
And I hope it is helpful for you, and you take up the comments, and also see to that you go for future presentations in a better form and without any fear. The event has met its aim or its goal. Thank you. Over to Hira.
Yes, sir. Thank you,
Will
sir.
add some more point.
Please, ma'am.
Yeah. All the best for all the participants. I really happy to for you to attend this and make your thing. If it is going to be happening or not, I really happy to see such young scientists to come up with your ideas, your work, which is being recognized here. And it will also be recognized soon in much more higher places. Keep working. Don't put your don't sit up on the within the four walls itself. Open your mouth and think out of box. And we faculties are there to support you in all means. Okay, you make use of it. Make use of your seniors, and make everything possible. All the best. Thank you.
Yeah, and one more thing, please don't get discouraged about what we said, oh, we are making a mistake or anything. We are just helping you come out in a better way. So take it in a positive way and try to make it work, okay? So always think about yourself in a positive way, no negative thoughts, be happy always, and strive further, okay? Strive to make it better. Thank you.
Thank you, ma'am. Thank you all judges for your motivation talk and for your constructive criticism. Actually, we students will take up these things and to the next level we'll try to push it up. First of all, I need to thank all the professors who have come to, instead of their busy schedule, they have matched up their timings and have come all the way to see the students work in the scientific arena and encourage them. I thank all Anusha ma'am, Udayamati ma'am, Ramya ma'am. Thank you all. And I need to thank Saramesh sir for initiating this event and the event coordinators too, who are all behind the scene. I thank all of the people. And the participants, each one of the participants here is actually a gem, because each gem has got its feature and the speciality, right? So in the same way, all the individuals they are unique in their way and they will become specialized in some field or another. So don't lose your hope and determination towards your goals. Every one of you is winner here. So all the best, and your results, winner and runner-up, will be out during the valedictory function. So let us break for lunch now and see you in the valedictory function, and even quiz before that. Thank you all. Thanks for the participation. Yeah. See you in the valedictory function. Over to Sai sir. Sai sir.
ஓகே வெயிட் பண்ணுங்க. அப்போ கனெக்ட் ஆகும். கனெக்ட் ஆகணும்னு சொல்லுங்க. கால் பண்ணு. அது கட்டி தான் முடிச்சுட்டு வரணும்.
Okay, Hira, we can close. Thank you.
Okay, Vignesh. Yeah, we can.